So our body is made up of tiny little cells. Within these cells, we have our own code, our genetic code. And this gives us the instructions to build proteins that regulate all the major functions of our cells. They tell the cells when to grow, when to divide, when to die. When we have mutations in our genetic code, it can affect how the proteins in our cells fold, and it can affect how they function. When proteins that control these vital functions do not work, it leads to disease. Some of the major diseases that we see with these types of mutations are cancer, tuberous sclerosis, epilepsy, and even autism. Tuberous sclerosis is a disease that affects about 1 in every 10,000 people. It causes non-cancerous growths in the skin, in the brain, in the heart, in the lungs, and in the kidneys. This disorder also has profound effects on the nervous system. It can lead to epilepsy, autism, intellectual disability, and developmental delay. Tuberous sclerosis is caused by a mutation in one of 2 genes. One of these genes encodes for a protein called tuberin. Tuberin is known to help regulate how the cell grows and divides. Tuberin regulates these vital functions by binding to another protein called cyclin B1. We hypothesize that mutations in tuberin might affect how it binds to cyclin B1, affecting how the cells grow and divide and leading to the diverse clinical presentations we see in the patient population. We are examining a number of mutations found in the cyclin B1 binding domain of tuberin that are seen in the clinical population. We are characterizing the different mutations in the hope of understanding the cause of the diverse clinical presentations within the group of patients with tuberous sclerosis. We are also working on identifying which mutations lead to the most severe clinical presentations. Currently, we are examining where in the cell these mutated forms of tuberin are localizing and how it binds to cyclin using a variety of advanced molecular biological techniques. Finally, we are making use of CRISPR-Cas technology in order to make cells that have mutant versions of tuberin naturally. So CRISPR-Cas technology was commercialized in 2013 and has very broad implications for research and for healthcare. It allows for us to induce mutations, turn off and on genes permanently within a cell. We're using it to make our cells express certain mutants of tuberin so that we can see how these mutations reflect in the cell in normal levels that you would see in a patient. Patients who express mutant versions of tuberin develop tuberous sclerosis and have an increased risk of many cancers. Having a better understanding of how such changes in such large proteins affect very fundamental biological functions such as cell growth and cell division will help us better understand disease and hopefully move forward towards increasing treatment options for these patients.